Now, speaking of future-proofing, if you will, and remaining agile, let's talk a little bit about how we're trying to decide which aspects of process to future-proof, and perhaps even from a regulatory compliance standpoint. What do you keep flexible while, of course, the clinical data needs to continue to evolve? So, Ray, I'll start with you, and then Todd, I'd like to hear from your perspective as well.
Great. A lot of this will be, again, I think it's a lot about process understanding and where your process is. And I think it means that with that process understanding and expertise and being the subject matter experts in your product and your process, it is identifying where it seems like the pain points are, from every perspective, from compliance to scaling to capacity to maybe even the phenotype of the cells that are produced. I think understanding that and understanding where the pain points are will allow you to understand where you will get the most value out of the work from the initial work that you're performing, so that you can future-proof your process. And it'll also allow you, again, to design sort of a process roadmap so that you can, as you have funding and time and opportunity to improve it, you now have a roadmap to be able to do that.
Sure. And Todd, I'd like to ask you the same question. Go ahead.
Yeah, this is an interesting one. And I think this is where perhaps autologous and allogeneic may have slightly different challenges. In the allogeneic space, of course, we're going to make many lots from unique allogeneic donors, but those lots, need to be representative of each other for the same product. So in that regard, it's viewed kind of from the biologic lens. So there is a strong focus on product quality and safety, and ensuring that every lot, is representative of the previous lot. So with that in mind, and I touched on this a little bit earlier, we really focus on the significant areas that cause variation to your drug product quality. So a few examples of that might be really understanding donor variation, how that might interact with your process, really understanding the genetic engineering components of your process and how robust those are and interact with the various components of raw material and process. Ensure that your supply chain is robust. We're using very complex raw materials, sometimes, viruses and genetic editing materials. And then finally, I would say where you can invest early in method development. It really is indicative of your, purported mechanism of action. And you're going to have a lot of assays early on if you can. And although some of those might reflect, in vivo, some might not, and that will come later once you have your clinical data. So I would put those together as high priorities, kind of on this towards a single cycle development if you can. That's more easily said than done. I think things that are flexible that one could take later on, although nice to have early, is the closed automated processing. So ideally, the process is fully closed and automated, but I think those things can be implemented with the right infrastructure later on in the development cycle.
Great, thank you. I want to address a question that came in from Rahul. They're saying, do we need stability studies for any change in the process during clinical trial or as a post-approval change? For example, if I use selection beads from alternate manufacturer, same markers, example CD4 or CD8, then do I need to reduce stability studies? If yes, why? And I would open that up to either of you or both.
I can answer that. At least I can give my perspective on that, and Todd, if you have any input as well. So I think a lot of this depends. Again, it's phase appropriate, especially in autologous. There's going to be a different answer for phase 1 than there is for phase 2 versus post-approval. But again, the way that the FDA views this is, a lot of it will come down to, as I've said previously is risk assessments, right? So when you are, let's take the example that Rahul gave, which is you're changing your bead manufacturers. Do they matter? It says, will it affect the quality of the cell therapy? If you can make a good scientific argument that they are made from the same material, it is the same way it's selected, these are the same exact clones of the antibodies, and you can show that it is exactly the same just from manufacturer A for manufacturer B, then you might have scientific justification from the FDA to be able to say you don't need to redo stability studies. But I think it really depends on the risk. Oftentimes, in my perspective, if you're doing a filing and have to redo re-engineering runs and things like that, there's a high chance that they will ask for it. But a lot of it's going to be based on your scientific justification and sort of phase appropriate risks that you will undertake.
Yeah, I would just add to what you said there, Ray. In regards to stability, do the risk assessment. Not all raw materials, I think, will fall under the same lens in that regard. For example, beads, as mentioned, versus changing an LVV manufacturer site. As an example, may be viewed differently in regards to stability, and the risk assessment will vet that out. I think the bigger part of the risk assessment is what's the change in this raw material as it relates to product quality, and that's going to be the heavy lens you're going to get from a regulatory point of view. And I would spend a lot of time thinking deeply about the data to support such a change.